Hello everyone, my name is Malcolm Burns. I'm the head of the GMO Analytical Unit and Principal Scientist based at LGC in the UK, and I'm here today to give a talk entitled Towards Detection of GMOs and Gene Edited Products, Status and Challenges. This is part of the CCQM celebration of World Metrology Day on the 20th of May 2023, and the theme for this year is Measurement Supporting the Global Food System. As way of background and context to today's presentation, LGC, we are a Life Sciences Measurement Testing and Research Institute, and we celebrated our 180th anniversary last year. We have a number of official national and international roles associated with food and GMO analyses, including being a national measurement laboratory. So we are the UK's designated institute for chemical and biomeasurement, and we work globally to harmonise measurement science. We also have the UK Government Chemist function, which is a statutory role underpinned by relevant UK legislation, which is to provide impartial and independent referee analyses on a food sample as part of official controls, for example, in cases of dispute between a food business operator and a local authority. And another example of one of our official roles is as the UK National Reference Laboratory, NRL, for GMOs in food and feed, pursuant to the relevant EU regulation. In terms of status for GMO analysis, conventional GMOs can be defined as organisms produced using recombinant DNA technology, typically containing DNA sequences randomly introduced from the same or other species prior to the adoption of the relevant EC Directive 2001 and 18. And we've got a reference at the top of the slide there. And indeed, the analyses of GMOs revolves around supporting consumer choice, recognising that consumers may not want to purchase food which contain GMOs. Within Europe, the traceability and labelling of GMOs is governed by the relevant UK and EU legislation, which dictate that products containing GM material must be clearly labelled. The traceability of GMOs is underpinned through the use of an adoption of a threshold level for labelling. And for those GMOs authorised for use for placing on the market in the UK or the EU, there is a 0.9% de minimis threshold level for labelling, weight per weight of a particular ingredient. Hence the reason why we look at trace level detection using DNA. And also there is zero tolerance for those GMOs that are not authorised for placing on the market in the EU and the UK for food or feed use. Hence, the successful labelling of any food produce is dependent upon a reliable, stringent and efficient way of quantifying GM. The largest common denominator in the global framework of GMO analyses are DNA-based methods, and indeed the majority of accepted methods of GMO detection and quantification are DNA-based. We've got a number of reasons listed on the screen here. For example, DNA is ubiquitous, it's present in the cells of all organisms, whereas, for example, proteins can be tissue-specific or expressed at different developmental stages of an organism's life. Quantitative real-time PCR, qPCR-based analyses is the current preferred DNA-based technique for routine GMO analysis, and this is underpinned by the EURL validated methods for event-specific detection of GM varieties which provide unequivocal target identification. On this next screen, we've got an example of where metrology is used to underpin some of the regulatory controls. And the figure shows the estimated mean and 95% confidence intervals for the GM content associated with, for example, food samples. Here we're looking at assessing the samples with respect to compliance against a legislative limit for labelling. Now, should all of these samples be unlabelled, i.e. they do not show GM as labelled ingredients, the only sample which would be considered non-compliant would be the sample on the far left of the slide, sample A, as the mean and associated 95% confidence interval is above the threshold level for labelling. There are a number of challenges associated with GMO analysis. We've listed two of the key challenges here. On the right hand side you can see a graph showing the total number of GM lines authorised for use within the EU, ranging from 2005 to 2021, going from a handful of lines up to above 90 lines. And indeed, that's one of the key challenges now, testing for the increased number of GMOs. We can capitalise upon common GM control elements and markers which are present in GMOs, such as promoters and terminators used in the transformation or transfection of GMOs. And indeed, these screening tests can be used to cross-reference results from an informative panel of screening markers to narrow down the number of occurrences or the likelihood of GM lines being present in a sample. The second challenge is testing for unauthorised or unknown GMOs, so a range of different GMOs not authorised for use in the EU or the UK, and such things as next generation sequencing are providing a good basis for detection of these unauthorised GMOs. The second half of the talk consists of looking at gene editing. We've got an ISO definition at the top of the screen here, whereby gene editing can be defined as a group of new targeted mutagenesis techniques that facilitate addition, removal or alteration of DNA sequences at a specific location in the genome. You may also heard of such things as new genomic techniques in the UK, precision breeding, synthetic biology, etc. We've got a list of example techniques there as well, including CRISPR-Cas nucleases. Now, gene-edited products are distinct from conventional GMOs, the latter of which can be defined as being created via transfection or transformation, incorporating larger pieces of foreign DNA into the host genome, often at random sites, frequently along with easily identifiable markers, for example, promoters and terminators we referred to earlier. Now, gene-edited products can generate different kinds of alterations in the genome, ranging from single nucleotide variations all the way, and also deletions and insertions, all the way up to many base pairs. There are a number of challenges associated with detecting gene-edited products. We've listed two of the key challenges here. The first is the technical detection of small sequence alterations. It is technically challenging to detect small genomic alterations, for example, single nucleotide variations, but it is entirely feasible that this can be done given prior information on the sequences of interest and the associated DNA flanking regions as well. Modern molecular biology techniques, including quantitative real-time PCR, digital PCR, and next-generation sequencing, currently offer the best potential for detecting genetic changes in an organism's genome. Now, whilst this is globally recognised, what these modern molecular biology methods cannot do when used in isolation and targeting single small mutations is provide unequivocal evidence as to the source of that mutation, be that as a result of gene editing compared to, for example, natural processes such as traditional breeding or natural mutagenesis. And that's the second key challenge for detecting gene-edited products, establishing what the source of a mutation is. Now, finally, there's a number of other challenges associated with detection of gene-edited products, aside from the two key ones described on the previous slide. We'll briefly mention a few of these. We do not have time to go into any depth in this 10-minute presentation. Terminology is one of the challenges there on gene editing. Gene editing is an umbrella term encompassing a range of diverse techniques and mode of action of those techniques. But this diversity is also mirrored by the plethora of the different terms and terminology being used, and there is a need to harmonise this terminology in order to afford good understanding and comparability of gene editing products. Quantitative estimation is also important, for example, when assessing compliance against a legislative threshold. However, the lack of current validated methods for detection and identification of gene-edited products currently preclude the effective establishment of a quantitative approach. Screening for gene-edited products is also a challenge for GMOs. We briefed upon earlier that we can capitalise upon common genetic elements which are typically associated with production of GMOs, for example, promoters and terminators. But for gene-edited products, this is challenging due to the lack of inserted elements as a result of gene editing activity. And finally, other challenges exist as well in terms of agreement and production of relevant reference materials and databases. as well as the need for further research and development into the possible utilisation of weight of evidence approaches for traceability of gene-edited products in the food and feed supply chain. So to conclude then, conventional GMO controls and analyses is underpinned by evidence-based research and innovation, but further challenges do await in terms of both the number of GM lines, the presence of unknown and unauthorised GMOs, as well as detection of gene-edited products. Now, whilst this provides challenges, this is not necessarily insurmountable, as if we look at the advent of traceability for conventional GMOs in the early 2000s, historically we faced a number of challenges then as well. However, through research, innovation and development of best measurement practice guidance on the world stage, for example as underpinned through the EU reference laboratory for GMOs in food and feed and supported through the ENGL, the European Network of GMO Laboratories, we were able to reach agreement on the measurement unit, how to estimate measurement uncertainty, how to validate methods, and what the minimum performance criteria should be for verifying the use of a method. So the continued contribution to the solution can include, for example, development of sound analytical science, the use of evidence-led research and development, the importance of validating methods and estimating measurement uncertainty, looking at the traceability of gene-edited products as underpinned by, for example, reference materials and databases. We need to be aware of the utilisation and importance of new techniques and technologies, for example, digital PCR and NGS, and continue to engage both at the national and international level to share best measurement practice guidance. So finally, just to acknowledge supporters and funders of the work we do at LGC for GMO analysis, including Department for Science, Innovation and Technology, the UK Government Chemist Programme, the National Measurement Laboratory at LGC, the Food Standards Agency, the Food Standards Scotland, and Department for Environment, Food and Rural Affairs. And finally, just thank you for your time for listening to my presentation today. Thank you.